Good morning, everybody. So I was, I guess as the academic on the team, I was asked to educate you about what we're talking about. And knowing that we tend to talk a lot as teachers, I was informed to keep it fairly concise. So I only have like 5 or 6 slides, so you'll be happy with that. I do want to thank the organizers first for bringing us all together. This is really my first time involved with the team, and it's great to already have met some of you, and both John and Deidre with great overviews of where the world is. So this is really a great start. So if I can get the slides up. I don't know if I don't think I have control. Oh, yeah, great, thank you. All right, so I really just want to be quite brief. You know, there's a lot of complication here, including terminology and sort of where our perspectives are on the subject, so I'm going to really just try and keep it focused. So we're talking about cultivated meats, pioneering safety in the next generation proteins. The other common way to refer to this field, at least the way we do in our program, is tissue engineering food. So how to create food essentially without the use of the animal. So the only part of the animal we ever need are the cells. Once we've made biopsies or isolated the cells, in principle, you never need the animal again. It's sort of free from animal husbandry, if you will, in livestock. So that's the principle. The cartoon I like to use to introduce the subject, and then I'll make it really complex on the next one, is here. And if you just kind of guide, and again, I don't know the background of many of you, so I'm going to get a little bit into the weeds with apologies to some of you, but if you just work your way left to right, this is the basic paradigm that we're talking about for this field of cultivated meats. We use cellular agriculture for our terminology, but left to right, pick your source of food, so the animal that you would normally consume, and then you isolate cells, and there are ways to do this either from biopsies from the animal or embryonic stem cells or other cell sources. There are lots of tools today to do this. But ultimately, for food, you really want muscle, fat, and maybe connective tissue cell sources to do this well. And ultimately, those cells have to be fully characterized and completely understood for the safety and related topics that we'll talk about on the panel. And then you go into the middle part of this cartoon, which is really how do you get a few cells that you isolate from a biopsy into gazillions of cells, right? So there's a lot of questions about what kinds of bioreactors you need to get the cells to multiply to the level you want. And companies, academic labs are all looking at different modes to do this, whether it's traditional pharma-based stir tank reactors, hollow fiber reactors, and many other iterations. This is a great area for innovation and continued growth. So once you've done that, you've got, in principle, enough cells to start making food, and then in that case, you go into the biomaterials or scaffolding side as we continue to the right. And there are many, again, formats for this. They could be fibrous, they could be sponge-like, they could be sheets, patterned, unpatterned. The list is very, very long and growing. The paradigm here is you want scaffolding that's going to be part of these foods, intersected with the cells we just painstakingly grew in the middle there, and everything has to be edible, food safe, and so on. So again, more topics for today's panel. And then finally, we go to the right, where you make products that in principle consumers will recognize and want to buy and consume. And we'll talk a lot about those products today as well. So that's the big picture in terms of what we're talking about. I should normally stop here and ask if that's clear. Any questions from my students today, but I'm not hearing anything yet. Okay, so then we'll move on and make it more complicated with apologies. It's not that much more complicated, but if again we go left to right, here we're inserting a bunch of other sort of sourced materials into this equation, right? So aside from the food animals and cells at the top left, you see at the bottom left, plants and microbes going into media formulations. They then go into that middle region again to grow lots and lots of cells. So we have an inroad there. And then on the outside, you see the top line goes to what we call unstructured food products. You know those like whether it's hamburger, sausage, et cetera, chicken nuggets. And on the bottom right, you see we go into structured, highly dense meats that you also are familiar with. So all this brings in sort of these other sort of definitions and topics we'll touch on in different ways. So back to our definition, cellular agriculture is this conglomerate. This is the way we look at it. This is how you're going to make essentially nutritious, healthy, safe, tasty foods of the future. Unstructured or structured, doesn't matter. This is all part of that equation, whether it's the upstream inputs and downstream products. We then go to, if I do this right, the red circles. That's where we get the alt proteins. You may hear that term a lot, or alternative proteins. So these are microbially produced products, but they're acellular, that are ingredients in a lot of these processes. They may be growth factors, they may be scaffolding inputs, they may be media components and other media components and so on. So those are the reds. And then in the blues, you see plant-derived materials. Those are already available and growing in popularity, although there's questions always about nutrition and additives, over-processing, and so on. So we're trying to look at where we can take the benefits of that and integrate them into the overall cell ag process for these future foods. So those are plant-derived materials. They're products from cells, but there's no real cells in there, right? So it's mainly the matrix the cell produces. And then finally, the integration of all this is really the cell-based ingredients that come from cell ag that in principle give us better quality foods at the output of this whole process. So that's what we're talking about as a panel. I hope this morning you'll hear different perspectives on the different parts of this process. This is an amazing emerging technology. It's nascent. I mean, we're talking the first even thought of this was 10 years ago, right, by Mark Post in the Netherlands. So we're talking just the fledgling parts. So we're trying to all do our job with a lot of context and a lot of care for safety, quality, and so on. So you'll hear about that. I do want to make sure we know that there are immense challenges ahead. So as you heard from the morning speakers, we need everyone in this room to play a part. And whether you see the value or don't, this is really important. So I summarized a lot of the challenges on this slide, whether it's keeping costs in reasonable levels, whether it's the scaling of the manufacturing of these products. We are nowhere yet to the scales that we need to get to. I talked about unstructured and structured foods. Unstructured, relatively easy to make, right? Structured, dense foods, it's a huge challenge. If you think about just fundamental physical phenomena when you grow cells, cells don't do well in dense environments. They can't get access to nutrients, and they can't get rid of their waste products. So how do you overcome that? Again, we don't have time right now, but there's lots of innovation coming here. So stay tuned on that as well. Safety, 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 bottom left, you'll hear a lot about that. None of it's been demonstrated to scale, but so far we see a lot of value in what we're doing. And also the idea and the concept with this technology is to try and move away from using antibiotics and other additives so you get much healthier, quality, safe foods. And that's, again, work in progress. Food taste and quality, I always tell this story. So we started, thanks to Deidre and her team, 2 years ago, we started our National Institute for Cellular Agriculture at Tufts. And one of the questions we could never answer until about 3 months ago was the following. If you take meat from a cow, you all know what that tastes like, and you know what it smells like, before and after cooking. There was no example, really, of taking cells from that cow, muscle cells, getting those cells immortalized, growing up a piece of meat or growing up a slab of fat, and then asking the question, does it even smell or taste anything like the real thing, right? So that was a huge sort of, I don't want to call it a barrier, but a question that we always had, and the students in the lab were really excited about this. So it took, I think, 1.5 years for us to get our IRB approval to allow our students to smell, cook, and taste all these things that we do in the lab. And I'm happy to say it's amazing because the quality, at least the fat that we smell and so on, is really not that different at all from what you see when you smell and cook a pig fat and so on. So I think we're often running in the right track. I would say even beyond that, we can now bring into that really detailed analytics and quality assessments, right? So we get the whole profile of fatty acids and other components in those cells and meats that we're making. We can cross-reference that to the native foods and really look in detail where in the process you want to be to match up the best you can. And not to go off on my long stuff too much, but if you haven't thought about this, the opportunity in this technology to have direct access to the cells for all these foods is completely a game changer. because you can induce those cells to take up, let's just say, healthier fatty acids or different components. So you can engineer in what those cells are going to be like in terms of nutrition, taste, and other qualities as we move ahead. So lots of opportunities. And then finally, GMO or non-GMO, our attitude in the lab is we do both, and we let consumers decide, right, based on safety, quality assessments through the agencies you'll hear from. So that's the overview of where the challenges are. I always look at a challenge as a great opportunity because we love the research. So we continue on, and obviously after today, if there's any interest in this, either come visit us in Boston or contact me, and I'm happy to give you more details. And with that, I think we all know why we're here for this topic, and you already heard it beautifully from Deidre, right? Whether you care about food security, quality, safety, animal welfare, food equity, sustainability, or population growth, we need these technologies as alternatives to what we have today. We have no choice. So we really need to keep pushing this forward. I did want the last slide to be an advertisement, not a real advertisement, but just to tell you about our broader program in one slide, and then I'll leave the podium. So as an academic lab, we obviously pursue the educational side. So we have the first of the kind in the world on both undergraduate and graduate degrees in cell ag, and we openly share all those syllabi and other lecturers with any other universities that want them, and we're also getting this out in the public. We have YouTube channel, we have a lot of other newsletters, we have seminars that are open to everybody. So if anybody wants to get on the mailing list, please let me know. I'm happy to say also for our visitor from Canada, the very first textbook just came out, and it's all co-edited between myself and 3 colleagues up in Canada. So very good international cooperation. On the upper right is what you heard from Deidre, which we're incredibly thankful for. We're now just entering the 3rd year of our USDA-sponsored National Institute for Cell Ag. We're now up to 7 additional academic partners. We have industry involved. We have seed funding for other initiatives, and just really going strong with, it's hard to get academics to share openly sometimes. This is a complete different approach where we all openly work together on joint problem solving for the different factors, co-publish together and so on. So it's off and running well. We'd love to add more partners, so please again contact me if that's of interest. We have an active industrial consortium at the bottom left, including some of the big companies in the space. We have ways to work with them in pre-competitive research, and then we just started a commercialization lab with some of the activities as well. So those are the ongoing activities, and I'll stop here and turn it over to the panel, wherever we go.
Thank you very much, Emmanuel Kesmali from Mahidol University, Thailand. I was so pleased. We're about to have a collaboration to discuss with Tufts University. So I'm so pleased to hear about your work. I just want to comment on some of the things about the needs. And one of the things with aging population and elderly has to do with the sensory. They start to lose all those. And some of these things are quite important for quality of life for them. Does this cell, is that a part of it where that could go, besides the nutritional design, for that to be more healthier profile, while they still can enjoy, let's say, a piece of meat?
So very, very interesting question. I think we're just at the beginning of exactly those kind of opportunities. Aside from the broader food needs and general nutrition and quality taste, safety, you have the opportunity now to include foods that are, I call them therapeutic foods, but foods that match an individual subpopulation, whether it's a medical need, whether it's a cultural need. Those are all things you can now do because of the technology we're talking about. I'll give you a very simple example that we published a year ago, which is we can take our bovine cow muscle cells, which are great for texture, taste, but you can engineer them to overproduce something like vitamin A. So you enhance the nutritional value it becomes, the cells become in principle more stable on the shelf because you have more antioxidants. And they survive cooking because they're protected by the cells themselves. So this is just one example of many iterations that are being worked on, both in our lab and other labs, where yes, you could improve that sort of flavor, taste, aroma, as well as the health and well-being for subpopulations.
Yeah, no, and just to add to that, I mean, like Dr. Kaplan mentioned, I mean, we are just at the beginning, so we're trying to replicate just your regular piece of chicken as much as possible, but the idea would be to eventually engineer it to fit a much more healthier product. But with regard to flavor, we are certainly also working with that, and we're also working with some of the flavor houses on the post-harvest bit in order to get that sensory taste as close as possible, or even enhance it if need be. So there's different aspects that we're working on that for sure. Any other questions? Yeah. Oh.
Hello, Koen Willekens from the Research Institute for Agriculture, Fisheries and Food in Belgium. I have a question about the nutrients and the minerals or carbohydrates you might use for the multiplication of the cells. What's their origin?
Sorry, I'm not sure I caught the last part. What is the origin of those nutrients? Is that what you
the
asked?
nutrients and the minerals, and yeah.
Right. So all that is the upfront media composition that you have to formulate to then support the cell growth and support the cell differentiation into muscle or fat. So that includes things like glucose, as well as vitamins, depending on the cell type, as well as all kinds of salts, and then growth factors. So this is all a portion specific to a species and cell type that you're trying to expand to create these products. And this is all where supply chains are going to be needed to fit all this as we move forward. Those supply chains don't exist yet. They're at the scale we're going to need. And that's active work out there in the community as well. So I hope that I answered your question if I understood it. Yeah, thank you.
Thank you.
I've got another one over here.
Hi, I'm Connie Weaver from San Diego State University, and kind of following up on the dialogue you had with Emorn about you're at the beginning to make decisions about where to go, and you don't have the same lobbying interests for a particular product, I wouldn't think. You can correct me if that's wrong. So you could make a lot of interesting decisions. Like I heard last week a seminar that said 40% of the food products around the world are halal and kosher. So you could not try to make pig products, instead go to some other animal source. But how do you weigh consumer demand for taste against feeding the population of the world?
Is that for me or someone else? Yes? So there's different ways to address that. It's a really good question as well. I think we've tended to start with the products, the food products most of the world consumes, right? Porcine, cow, chicken, those are kind of the favorites. And then when we go off the coast of Massachusetts and catch fish, we catch a mackerel or a bluefin tuna. That's the cells we use from fish to start. But I think you raise a really good point, which is, again, another novelty for this technology. And I don't know of many companies other than Vow in Australia doing this, where you can really recover a cell from any animal on the planet and create food from that, right? Because once you've got the initial biopsy, as long as you can grow those cells in quantity, you can produce a food from that, right? So think of the possibilities of maybe 10 years from now, maybe 50 years from now, where your food palette is going to be completely transformed. Instead of the dozen or so traditional meats and fishes you have in the supermarket today, you'll be able to have, in principle, 1,000, right? Because there's nothing preventing this broadening of our food tastes and opportunities. How is that going to happen? That depends on consumers, depends on companies, depends on safety. But but that's a game changer again for this technology. A lot of work to do to get there, but we're getting there. So, again, I don't know if that answered your question enough, but those are decisions, and companies ultimately will make those decisions. We'll do the research, publish the research, share the cells. We openly share our cells that we isolate and characterize, but ultimately it's going to be the consumers and the companies to make those choices.
Yeah, no, from a startup perspective, one of the reasons that Upside decided to go first with chicken was, of course, we did a consumer survey, and for the most part they said, oh, chicken would probably be the most, one of the things that is more widely accepted to start off with. And I mean, at the end of the day, we're trying to eventually become a profit company to keep on going. So that was one of the reasons in our case. But certainly, other people are doing seafood and beef. There's several beef companies out there. So certainly, and we're working towards doing all of those as well. There's one back there.
Hi, Tia Rains with Ajinomoto. Thank you for your presentation. It's fascinating work, and I understand that we're at the beginning of the technology phase, but I think we had a lot of learnings from the whole GMO topic, and I'm just wondering, Tufts has a great nutrition communications program. Have you thought about getting ahead of the consumer acceptance piece by having a more proactive consumer campaign so that when the technology is ready, that people aren't fearing the technology and that their acceptance of the products will be high?
Yeah, thank you for that. So absolutely yes, and it's one of our core 8 pillars in our USDA Institute for Cellular Agriculture. It's consumer acceptance questions. So there's a couple of faculty colleagues, Sean Cash and Nicole Tishner-Blackstone in our School of Nutrition, and they are very actively working on exactly those questions. They run consumer surveys. They do it in collaboration with our other academic partners in the program, and those will get published as the data warrants, and there are some publications coming on this. They started with what's the best name to use for all this, because names carry a lot of weight, as you might imagine, and then the question is, would you consume it, and if so, why or why not? And then we go on from there. So yeah, that's all underway. Happy to talk more about those things. Thank you.
Question on Ralph Fritchie with the National Aeronautics and Space Administration. Question on when you foresee this being a commercially viable alternative to mainstream meat sourcing for the general population.
Yeah, I mean, again, we're still at the beginning right now. There's 2 companies here, at least here in the U.S., Upside Foods and Good Meat, that have received their no questions letter and their grant of inspection. And of course, you're not seeing our stuff in the grocery stores yet. We do have Upside Food has a restaurant that they're providing their product out there in the Bay Area. And while Good Meat has another restaurant out in Washington, D.C. And so that's really the way that we're trying to put our product out there. Granted, it's still not a $10 or even a $15 plate of chicken. To tell the truth, if you go out there to the Bay Area, it's about $150 per plate. So it's still far out of the reach of the typical consumer. I mean, I'm going to wait till there's coupons, quite honestly, for that. But really, more to your question. I mean, right now the scale up is really the issue right now. I mean, just trying to get cheap or media reagents in order to do that. Trying to get the equipment, the bioprocessing piece as well. There's just not enough media to go out there for everyone to use it to scale up currently to eventually be able to compete with the conventional products. That's just unfortunate at this moment. With regards to the cost, one of the things that I think all startups are also dealing with is just trying to get something that's food grade equivalent at a good price. I mean, a lot of these cell media reagents are either technical grade or farmer grade, and so in order to make something food grade, we have to take on the cost of ensuring that there's no pesticides, there's no food allergens, and at the same time, even farmer grade, which is a bit better than technical grade, it doesn't account for food allergens or things of that nature. But I guess more directly to your question, when could we see that? I mean, I'm still thinking it's maybe 20 years out, to tell the truth.
Thank you, Ania Leon from Mesoamerica. My question is about these things about cost benefits, because, for example, in Mexico, there's a lot of people working to produce proteins from insects, and it's very, very expensive. So I want to know, for example, for our countries, what could be the cost, if you have a perspective about it? Of the protein, a grain of protein, cultivated meat, related with, because for us in the south of the planet could be a very good opportunity, but it is expensive.
No, certainly. I mean, with regards to just alternative protein in itself, we've got, of course, fermentation or precision fermentation technologies that are working on that to just produce the protein in itself. Again, I mean, it's still fairly expensive compared to the conventional products, of course. As far as cost, I just don't know at the moment, but it's certainly not something that you'd be able to get at a cheap price at the store, unfortunately. Yeah.
Maybe I can just add a little bit of hope to that. So again, this is ongoing technical innovation in many places around the world. So I don't want to minimize this. There are huge advances just in the last few years. Examples include looking at other host systems, host cells to generate these proteins, right? So, traditionally you use things that came from pharma. Now people are looking at all kinds of other cell sources, whether they're plant-derived, microbial-derived, and otherwise, to bring the cost down. Number two, media ingredients that was mentioned. People are looking at much better ways to have lower cost sourcing. Some of these are agricultural sort of resources that are already going to waste but are repurposed for this to immediately drop the cost. And then I would call it a more GMO approach, which we have published recently. You'll see a paper literally this week, is have the cells that you're using to make these alt proteins and even the cells make be self-primed. So you don't need expensive ingredients. The cells make the ingredients to get themselves to make the product you want. And this is a game changer as well, because this hasn't been done before. And it works, right? You still have a lot of work to go. But my point is there's a lot of innovation happening out there. And it was only recently that cost benefit was considered important, because this is for food as opposed to pharma. And now that people are working on it, you're going to see great strides in the coming 5-10 years. I'm very confident. Okay.
Yeah, maybe if I could just add to that, as the technology builds, there are going to be the infrastructure built up around it that helps support the companies. And the example that I can think of right now in Canada, there's a company that started out as a cellular agriculture company wanting to make meat, but instead have pivoted to making growth factors specifically for cellular agriculture. So you'll get these companies that come up that find better and cheaper ways to make the growth factors that are used to go into the medium than the ways that the companies have been maintaining them. And so as those things start building up around the industry, the cost will eventually fall. It's just the infrastructure has to be there.
Let's take one more question before going into the regulatory and safety piece.
Yeah. Thank you. My name is Sushila. I'm from ILSI, Southeast Asia region. And my question is, we sometimes drive hundreds of kilometers to a specific farm to buy meat, because that meat is defined by the culture of the farm and the environment. Can you stimulate that in cell cultured meat?
Sorry, can you repeat that?
Well, meat from a specific farm, sometimes it's defined by the culture of the farm and the environment that the animals grow in. So this is very specific, and people do drive, thousands, well, hundreds of kilometers just for a specific kind of meat. Like I come from Australia, Warwick is extremely famous for meat. People drive there hundreds of kilometers just to buy beef from a certain farm there, because the texture is great and the taste is great as well. So I'm just wondering whether in this, can we get that? Can you simulate that in your cell culture environment?
Eventually, that's the goal. As David mentioned, I mean, the easiest thing right now is to do unstructured meat. So think of a hamburger patty, that sort of thing, as opposed to structured, more like a steak. Yeah, I mean, we're certainly, that's the way, certainly the way that we want to go in order to get the taste profile and the sensory profile. But yeah, I mean, the hope is to eventually be there.
Yeah, I'll add to that as well. So there are already companies making Wagyu, right, as an example. So
yes,
yeah,
yes.
it's technology's there to do that. But what I think what you're going to see, and again, it's part of our USDA program, is to bring AI and omics technology into this whole field in a level that's never been done before. Right, and so if we go to that farm where you want to get your meat, if we can look at the ingredients, we can sort of characterize them in detail and then emulate that in what we feed the cells from that cow that we isolate by biopsy in the laboratory and then in a company. And in principle, there's nothing limiting sort of replicating that kind of quality, specialized meat from that location under those conditions. It's just going to take a lot of data and a lot of characterization, and a lot of tool building, and a lot of people to do that in a very systematic way. And that's one of our long-term goals, is to start to build that sort of infrastructure. And just to wax too poetically, apologies, my vision long-term, right, is if you know what the food is you want, right, it's kind of working backwards from an omics AI platform. You know everything that's in there from an analytical standpoint, right? Fatty acid composition, subtle flavors, everything is characterized. And then you can feed back to, okay, what cell do we put into the process? What do we feed it? And you'll know all that because you've characterized all that up front. So then it becomes just, computer-driven insight into make the food you want to come out the pipe at the other end. Do I think that's feasible? Not today. But I do think it's feasible in the long run. It's going to take a lot of effort, a lot of resources to make that, and a lot of people to make that happen. But that's the holy grail that we want to get to. And then you can get the food you want from wherever you want.
Can I just ask a mini question here?
Yeah.
How about the human touch? Because this specific farm, for example, we go there because the farmers treat their animals very well, and there seems to be something special about the meat from there. How would you bring that AI, maybe the farmer's wife, the way she pets the cow. I don't know what she does, but who knows. Yeah. But they advertise that actually,
Yeah.
So
No, very fair question.
Yeah.
I think ultimately after whether they're massaging the cow or playing the right music, I don't know that. But ultimately that's going to be reflected in that cow's composition, cell ratio, marbling, fatty acid composition, again, those are all things we can distill down to the science. And once we know that, in principle, yes, we can replicate that. Again, I'm not pretending we're there today or going to be there in a few years, but that's where we need to go with this field. And this is a field you can get there with. That's the amazing part of this. That's all able to be done with the right technological advances, okay? Thanks. I'd love to go visit that farm sometime, okay?
Yeah, no. I can say that some of our scientists do play a lot of music in our lab, so maybe that might have some sort of effect.